For today's lab, we are going to go over kingdoms Bacteria, Protista, and Fungi. Prokaryotes are going to be the first organisms that evolve on the planet, and it's believed that they evolved around 3.5 billion years ago. And there are two distinct groups of prokaryotes, and they are going to be bacteria and archaea. Now, prokaryotes are thought to have lived and evolved all alone on the Earth for about 2 billion years. They are basically found anywhere on the planet and can survive in habitats that are often too cold, too hot, too salty, acidic, or alkaline for most eukaryotes. If you look at all the collective biomass of every prokaryote versus all the biomass for eukaryotes, the prokaryotes are actually going to outweigh eukaryotes by at least 10 times. So they are enormously important and a big group of organisms on the planet. They also are going to be important because they cause about half of all human diseases. So prokaryotes are going to be found basically everywhere, including on our bodies. So we have what we call a microbiota, which is the community of microorganisms that live on and in our bodies, and they help us to do many things. For example, they help supply essential vitamins, they extract nutrition from some food molecules that we would not be able to extract on our own. They also help decompose dead skin cells on our surface of our skin. They also are going to be kind of a first line of defense against disease-causing intruders because they're going to take up the space and they do not want to have other bacteria come in and take over for them. And so they basically are on the surface of our body guarding and kind of defending their space. Prokaryotes are also going to be extremely important in the environment because they're going to help decompose all dead material on the planet, so including dead bodies. If we didn't have things like prokaryotes, then waste would just build up. But it's really important for us to be able to return all the vital chemicals that are inside of dead organisms. Now, for the structure of prokaryotes, we've already talked a little bit about this, so it's important to just remember those main things, that prokaryotes are not going to have membrane-bound organelles, they're not going to have a nucleus, they do have a plasma membrane, and they usually are going to have a cell wall that is around the plasma membrane. Now, one thing about this group, the prokaryotes, is that they're extremely diverse, and you can find lots of different kind of forms. They can look very differently. About half of them will have some type of flagellum, which are these tail-like structures. They look very similar to what we would see kind of on a sperm cell, but these are the flagella. They are going to allow for bacteria to be mobile and kind of move around. There are three common shapes of prokaryotes, and they're going to fall into these three categories. The first are spheres, and they're going to be called cocci. The second is called bacilla, and these are going to be rod shaped, so kind of look like a hot dog. And the last group is going to be a spiral, and we call them spirillum. So make sure that you are able to remember the names of these three different groups. So we tend to go with the kind of the name that's in the black kind of bold letters. So cocci, bacilli, and spirillum. We're going to spend most of our time on kingdom bacteria of the prokaryotes, but I did want to mention kingdom archaea because they kind of are very interesting organisms. They are going to be abundant in lots of different habitats, and some of the habitats that they'll live in are places that few other organisms can actually survive. So three of these kind of very extreme environments are going to include extreme thermophiles, which are heat-loving archaea. There are going to be extreme halophiles, which are going to be salt lovers, and they live in extremely salty conditions. And the third group are going to be these organisms called methanogens, which live inside the guts of organisms or live on the bottom of kind of lakes. And the ones that live inside of the guts, they will give off kind of methane as a byproduct. So now we're going to go into kingdom bacteria, and we're going to start looking at the different groups. Now, kingdom bacteria is going to be very diverse. The types are going to fall into anaerobic, where they do not need oxygen, to aerobic, where some of them are going to need oxygen. They often will have symbiotic relationships with plants and animal hosts, and a symbiosis is going to be kind of a mutually beneficial relationship between two organisms. So one of the main ways that you categorize bacteria is based on their cell wall. So all bacteria are going to have a cell wall made out of peptidoglycan, which is going to be made of sugars and amino acids. Now, there are two major categories of bacterial cell walls. And so we have what we call gram-positive and gram-negative. The big difference between these two are kind of the thickness of the gram-positive and gram-negative's peptidoglycan wall. So if you can see on gram-positive, they're going to have a very thick peptidoglycan wall. Okay, so they're going to have this wall that's going to be able to kind of protect them. The gram-negative are going to have a smaller peptidoglycan wall, so it's not as many layers, and it also is going to have an outer membrane that's on the outside of that cell wall. This outer membrane is often toxic and can cause, if it's ingested by humans, kind of sickness like food poisoning. Now, this is really important because this will come up on the practical, but the big difference between gram-positive and gram-negative is that they are going to stain a different color. So that difference in the thickness of their peptidoglycan wall will stain differently depending on what they have. So gram-positive bacteria are going to stain purple or kind of look this bluish color, and gram-negative bacteria are going to stain kind of this pinkish reddish color. So if you see bacteria under a microscope, and remember, bacteria are very tiny and they are either purple or pink, you're going to know that it's going to tell you what kind of bacteria you have, if it's gram-negative or gram-positive. So please, please remember which one is which. So gram-positive is purple, gram-negative is pink. Unfortunately, they both start with P, so that won't help you. But if you can just kind of remember which one is which, that'll help you for your practical. Now, for this first exercise on page 120, what you're going to do is look at these three kind of images of bacteria. You're going to describe what the three different shapes look like, what color they are, and then say whether they are gram-positive or gram-negative. One thing that prokaryotes, and specifically bacteria, do is that they can accumulate on surfaces and become highly organized, and this is going to be called a biofilm. They can consist of one or several different types of prokaryotes, so you can have different types of bacteria, and they can form on almost any material, including rocks, organic materials like living tissues, and metals and plastics. Now, the image that we have on this slide are actually going to be the biofilm known as dental plaque. And so this is going to cause decay for your teeth. So if you don't brush your teeth very often, you can have dental plaque kind of grow out of control and it would start to cause tooth decay. Now, other biofilms are going to be kind of a common type of kind of human disease, and so they can cause different types of issues. Now, for this next activity, what you would do normally is you would take a scraping of your teeth. You take a toothpick, scrape your teeth with the toothpick, and then you would put it on a slide with some methylene blue. Now, in this slide that we have for you, you can see cheek cells, but you can also see these really little dots, and those are actually going to be bacteria. They're specifically going to be a type of streptococcus, and so coccus, think that is going to be the circular or spherical cocci. And what these bacteria do is that they sit on your teeth, they make this biofilm, and they are going to perform their normal metabolism. When they do that, they start to produce these acids, which will actually wear and tear on your teeth and enamel, which can then cause cavities. Now, the next activity, we're going to start talking about photosynthetic bacteria. Now, this image is going to be something that you want to be able to recognize by the image. So this is going to be Osculatoria. You would need to be able to look at this and be able to say that this is cyanobacteria, so it is a prokaryote, and it also is photosynthetic. Okay, so on your lab pages, it'll ask you some questions and you can answer some questions about Osculatoria. You will not need to know the name Osculatoria, but you would need to know that this is a bacteria, because this does look very similar to algae, but it's not. Now, this third activity with bacteria has to do with antibacterial properties of different household items. So what we do for this activity is that we have these two plates that have agar on them. And agar is a growth medium. We then separate the plate into four, and we are going to expose the plates to two different types of bacteria. So one plate is going to be exposed to a gram-positive bacteria, Streptococcus aureus, and the second plate is going to be exposed to gram-negative E. coli. After they have been exposed, we then put a little disc of paper that has been soaked in different types of antibacterial cleaners. So we have Bactine, Dawn, Lysol, and Clean and Clear. Those little pieces of paper that have been dipped in those different cleaners are then placed on the plate where the bacteria are. Okay, if there starts to form a clear zone, and so you can see a clear zone, so the black thing in the middle is going to be where the disc of the cleaner is, and then if you have a clear zone around it, that means that there's an inhibition of bacterial growth. So the antibacterial cleaner is actually starting to kill the bacteria. Now, on the next slide, we're going to go over some results to see kind of what would have happened. And so these are pictures from old labs that we've done. So here we have the Petri dishes that have sat over kind of a week and have been in the incubators to allow for growth of the bacteria. Now, we have kind of A, B, C, D written on the plate, and those are going to represent the different cleaners. And so the cleaners are written down here. So we have them, and we can then look at what cleaner actually worked. So again, we're looking at kind of the clear space around the disc and seeing if there is actually going to be inhibition of growth. So the top plate is going to be our gram-positive, and our bottom plate is going to be our gram-negative. So we want to see which of the different cleaners is most effective and if they work better than on one type of bacteria than other. And so I think what you're going to see is that gram-positive seems to be inhibited more than gram-negative, and there are certain cleaners that don't even do anything for the gram-negative, like Bactine and the Lysol, which is kind of surprising because Lysol is supposed to be effective. So then you can take kind of these results and come to some conclusions and answer the questions on page 122. Now, normally what we would do the week before we do this lab is we would have you guys take one of these agar plates and put your finger and actually touch the agar and transfer some of the bacteria and funguses that are on your hands. Now, when we do this, we then wait over a week and let everything grow, and we often are going to have a lot of growth from your hands. It doesn't mean that you're dirty, it just means that you have a lot of bacteria and fungus on your hands, which is normal. So you can see different types of bacteria and fungus on here. The difference between bacteria and fungus are kind of the way that they appear on the plate. So bacteria tend to be smooth, and fungus tend to have kind of these rougher and fuzzy looks. This is going to be really hard for you to actually see any big difference, but the main thing to take away from this activity is that you are covered in bacteria and funguses. So we're now going to move on to kingdom Protista, and so this group is going to have some interesting organisms and is going to contain kind of a big group of diverse eukaryotes. So protists are going to be the oldest known eukaryotes on the planet, and they're thought to have evolved from prokaryotes about 2 billion years ago. They are going to be the predecessors of kind of a variety of the modern day protists and ancestral to all of the other eukaryotes, including the plants, funguses, and animals. So the protists are going to represent some of the oldest groups of the eukaryotes. Now, this is a very interesting group because normally taxonomy is going to tell us something about the relationship between these different organisms. But in the protists, that's not really how this works. Now, the protists are very interesting, and we keep on finding more and more new information about them, and their kind of relationships to each other keep on changing. And so the protists are actually more of this catch-all group that basically don't belong in any of the eukaryotic groups, including fungus, animal, or plant. So the protists are generally going to be unicellular, although there are going to be some that are multicellular. Now, one mark of the protist group is that there's extreme diversity in almost every single facet of their lives, from kind of how they obtain their nutrition to where you find them to their anatomy. So there are going to be multiple different types of protists. We have photosynthetic protists. Some of these are going to be in this group that we call the algae. This will also include some cyanobacteria. So there are some cyanobacteria that are actual bacteria, and there are other that are going to be considered protists. Another group are going to be heterotrophs. So they're going to acquire their food from other organisms like us. So they're going to eat their food in that way. So some of them are going to be fungus-like, where they will actually absorb kind of molecules, and others are going to be parasitic. So some parasites are going to be like this trypanosome that we see kind of in this center picture. Let me just draw an arrow. So these are trypanosomes, and so they are going to be parasites. And a parasite is going to derive its nutrition from a living host, and it's going to harm that host in that interaction. There are also going to be other protists that can go back and forth, and they're called mixotrophs. So they can do photosynthesis when they need to, and they can be heterotrophs when they need to. So they're not kind of stuck in any one particular type of metabolic pathway. So protists will be able to live in a diverse number of habitats. The common theme is that they have to be moist. So many of the protists will live in actual water, in the ocean, lake, or pond, river, place like that. Others will be found on land, but they have to be somewhere where there's damp soil or leaf litter. And other types will be symbionts, which means that they're going to live inside the body of their host organism. And a symbiont means that it's going to be a positive relationship for both the host and for the protist. Now, because classification with protists is so messy, there aren't really kind of organized in a way that's telling us anything about phylogeny, but more kind of just how they have similar characteristics. So I want you all to be thinking about the practical when we go through these labs. And for the protists, you're going to need to be able to answer these questions for the different groups. Okay, so make sure that when you are studying, I would suggest making note cards and have an image of the different protists that we go over, and then on the back of it, you could have some of this information on there. Okay, you're going to want to know where they're found. Are they free living, or are they parasitic, which means that they live inside another organism? How are they getting their food? Can they move, and how do they move? Are they multicellular or unicellular, and do they have any unique structures? And I'll try to point those out when we start going through them. Now, this activity 5 is going to be going through the diversity of animal-like protists. So there's different groups of protists. There's some that are more animal-like, some that are more plant-like, and this first group is going to be on the more animal-like side. Now, this is going to include different protists, and they're kind of grouped based on how they move. So we're going to talk about amoebas that are going to use pseudopods. We're going to talk about ciliates, which are going to have cilia, and we're going to talk about flagellates, which are going to have flagella. So this is the description of the amoeba that's in your lab manual. And so you're going to want to make sure that you understand kind of their little brief synopsis, and I've highlighted some important things. So first of all, amoebas are going to be unicellular, and they are going to move by cytoplasmic streaming through these structures called pseudopods. So pseudopod is going to be a very important structure. Okay, they are going to obtain their nutrients via eating. So that's phagocytosis means they're eating, so they are going to be heterotrophs. Okay, and they are also going to have kind of food vacuoles that will have these enzymes for digestion. They will also have a special structure called a contractile vacuole, which will help expel water from their bodies. Okay, and so you're going to generally see them dyed kind of a pinkish or bluish, and they look kind of all different shapes, and you can mainly see their nucleus in the center. And so this is going to be what the amoeba would look like. On this slide, you're going to have a video of amoeba using cytoplasmic streaming and kind of making those pseudopods. So make sure that you are aware of what these look like when they are moving. This next group is going to be called Paramecium, and they're going to have cilia. So they're called ciliates. They have these little tiny hairs that are going to surround the entire body of the cell, and they are going to help them move in their environment, but also aid in feeding. Paramecium are not photosynthetic. They are going to be heterotrophs, so they're going to eat the food. And if you see these, I'm going to want to point them out compared to Euglena, because sometimes people get confused between this and another group called Euglena. These ones have cilia and they do not do photosynthesis. They're heterotrophs, so they don't look green, and so that should be kind of one of your key identifiers. This is going to be a video of how Paramecium moves, so make sure you take a look at that. This next group is really interesting, and they're called Trichonympha. They're going to have flagella, and so they have these long hair-like structures. Flagella are longer than cilia, and they're going to look similar to what a sperm looks like. So, sperm have this structure and then a flagellum, and so the flagellum is that long tail-like structure. Now, Trichonympha are going to live inside the guts of termites. And so in lab, normally what we do is we take termites and you squeeze out some of their fluid from their anus, and you can actually look at it under a microscope and see Trichonympha. Trichonympha are going to be able to digest the wood for the termites, and so this is considered a symbiotic relationship. Here's going to be a video showing you how the Trichonympha move once they've been taken out of a termite's gut. This last group of the animal-like protists are going to be called trypanosomes, and they're going to use flagella. They are also going to be parasites. And so what we're seeing here is actually a slide of a human blood smear. And so we can see the red blood cells that I've highlighted, and the frilly-looking pink organisms are the trypanosomes. Okay, so they can have some trypanosomes that live in fresh water and are not parasites, but the ones that we talk about in the lab are mostly going to be parasites. They can cause two different human diseases. One of them is African sleeping sickness, and the other one is Chagas disease, and so they can be pretty bad. The way that I will always tell you to recognize this one is that you will see red blood cells and then the trypanosome. So if there are red blood cells, it has to be trypanosome because it's a parasite. Now the next group we're going to talk about are going to be the plant-like protists, and they're going to be doing photosynthesis. So they're often going to be green. Some of them on here are going to be colored other colors because of art reasons, but they're going to do photosynthesis. So this first group is Euglena, and Euglena can sometimes look like Paramecium, but the big thing is that they're going to be green, they're going to have a flagellum, which is this long tail, and they're going to have this red eye spot that you can see. Okay, they do photosynthesis, but they will sometimes also do kind of be a heterotroph where they'll eat food, but in general they are going to be photosynthetic. Okay, so they're considered autotrophs. This is going to be a video of watching Euglenas move through water. The next group are dinoflagellates, and these are going to be a very important group in phytoplankton. They're going to provide a lot of food for marine ecosystems and be extremely important. They are going to be photosynthetic, And they often have kind of these plates on their surface that kind of make them look like they have armor on. They also can be potentially deadly when they kind of explode and have way too many of them. They cause these blooms, which can cause red tides, and they also can cause kind of bioluminescence. So here is an image of kind of the beauty of the bioluminescence. So this is actually happening down in San Diego right now. You can see the bioluminescence. So some of the dinoflagellates will give off this bioluminescence at night. You can see it. But there also is kind of a deadly side to this. So they can cause these red tides, which you can see in the bottom picture. And those will actually kill a lot of fish and other sea life. And this is when they just have kind of explosive events of breeding, and there's lots of them in the water. The next group are diatoms, which are going to be another component of phytoplankton. They are going to have silica cell walls, which is the same material that glass is made out of, and their bodies actually will remain in the environment much longer after they've died. And we actually use something called diatomaceous earth, which is a filter material that is made out of the skeletons of diatoms. Diatoms also will, they form their food and store it as oil instead of carbohydrates, which is kind of interesting. And so oil from fossilized diatoms will actually contribute to the fossil fuel supply. So we have a diversity of diatoms on here. I find that diatoms are absolutely beautiful, and this is one really beautiful example of one. It has this fan-like shape, and again, this is a diatom. The next group are going to be green algae, and the green algae are going to be kind of in both freshwater and marine environments, and these are thought to be kind of a precursor to what is going to end up being kind of a plant. So they're kind of in a different group, but they do have kind of precursor to the green plants because they have kind of the same chlorophyll. Right, so this first group we're going to look at, which are these little balls, are going to be volvox. And volvox are colonial protists, so when we're looking at these balls of green, that is actually hundreds or thousands of little cells. And each one is going to have their own flagellum, so they're going to have flagella as a means of locomotion. Now they live in this colony, and sometimes they'll have little daughter colonies that will arise within them. And so when you see this kind of larger green circle with little green circles inside of them, those are actually going to be the little colonies. On this video, you're actually going to see some of the volvox, and they move really beautifully in the water and kind of circle and spin around. Now, what I want you to do before you go to the next slide is to try to go through all of the different information that I just gave you and try to fill this out. Once you've finished looking through for the answers, you can then move on to the next slide, which is going to have the answer. Okay, so here are going to be what you should have found for each of these different groups. I want to just point out that I put not applicable for diatom locomotion. Most of them don't move, and we didn't talk about it, so I wouldn't worry about diatom locomotion. We're now going to move on to the last kingdom for this week's lab, which is going to be the kingdom that contains fungi. And so when you think about fungi, you're going to mainly think about mushrooms, but there are a whole lot of other types, groups that do not have mushroom shapes, and we'll talk about some of those. Now, some of the characteristics that are going to be in this kingdom are that they're all heterotrophs. So many people think that mushrooms and funguses are plants. I know that a mushroom is found in the produce section and it seems like it's a plant, but it actually is not. They are completely heterotrophic. They do not perform photosynthesis, even though I chose a picture where they're glowing green. That is not chlorophyll. They're actually glow in the dark. They're going to be decomposers, so they are going to live on dead material and kind of extract the nutrition from it. They also can be parasites. Now, when they digest their food, they actually sit on top of it, and they're going to send these things called hyphae down into the food and start to secrete digestive enzymes that will then break down the food, and then they'll kind of slurp it up back into their bodies. Okay, they're going to have cell walls that are made out of chitin, and this is interesting because chitin is also the same material that the exoskeletons of insects are going to have. And so fungi is actually more closely related to animals than it is to plants, if you look at kind of the genetics of it and also some of these shared characteristics. Now, one thing about funguses is that they have kind of a sexual and asexual phase of their life cycles that can get kind of confusing, but we'll kind of go through that on these slides. So this kingdom is going to be divided into 4 phyla, which is going to be the next classification that we go over. So remember, we have domain, kingdom, and phyla. So the 4 phyla are going to be classified based on the kind of spore type they have. So Chytromycota has something called a zoospore, Zygomycota has a zygospore, Ascomycota has an ascus, and a Basidiomycota has basidium. And you can actually see those in this tree with the different images. So funguses can be either multicellular or unicellular, but we're going to mainly talk about kind of what they look like when they're multicellular. So when you're looking at a mushroom, part of the major structure of the mushroom is actually below the ground, and the mushroom is something called a reproductive structure. So the part that we see or that we eat is what they use for reproduction. The rest of the fungus is actually underground and kind of invading whatever it's sitting on. So they have this structure called the mycelium, which is going to be composed of lots of hyphae, which are kind of these tube-like structures that are going to branch and kind of invade different areas. When the mushroom is wanting to reproduce, it will then produce this reproductive structure called the mushroom, which will then have spore-producing structures on the underside. Now, like the protist, you're going to want to be able to answer all of these questions for the fungus groups. So make sure that you know where they're found, what are the names of the reproductive structures, how did they get their food, and a hint for this one is they are all decomposers. So they're going to break down dead and dying material, and then also know a couple examples of them. The first phylum is going to be Chytromycota, and this group is going to be found throughout soils and in lakes. It is also going to have some decomposers, but others are going to be parasites. And one problem with this phylum is that there are becoming kind of increased widespread infections of amphibian populations, and it's killing out lots and lots of amphibians. Some chytrids will actually live in the digestive systems of cattle, and so in that case they won't necessarily be a parasite but a mutualist. And this is thought to be the first phylum to evolve, and they're going to have zoospores as their reproductive structure, and so they have flagellated, or have flagellum that are going to allow them to swim in water. The next group are phylum Zygomycota, and this is a familiar one. You've probably come into contact with it because it is the type that causes bread mold, and that's called Rhizophis. This is going to get its name from the reproductive structure called a zygospore. And so what happens is you kind of have the fusion of 2 different mating types coming together and forming this kind of interesting structure where you have kind of a center part and then these 2 different hyphae from the other groups. This next group is called phylum Ascomycota, and they are going to be the sac fungus. They are going to have spores that are produced in sacs called ascus, and you can see in ascus, I outlined it and highlighted it in that yellow circle. So these are going to be a very versatile type of fungus, and they can decompose lots of things, including paint and jet fuel. And they also are going to be some delicacies like truffles and morel mushrooms, which can be very expensive. They also are going to have some of the types of yeast that are going to be used for both baking and alcoholic beverages. Another important member of Ascomycota is Penicillium, which produces the antibacterial product called penicillin, which was actually accidentally found by Alexander Fleming. And penicillin was the first antibiotic ever discovered. The last group of fungus is the club fungi, which are called Basidiomycota. And many of the mushrooms that we eat are going to be found in this group. So what the reproductive structure is called a basidia, and they actually grow in the strips under kind of the mushroom cap on these things called gills, and I'll show you a better picture of that. There is a very enormous diversity of these, and I picked out some really cute looking ones, or some of them are actually kind of horrifying, but these little ones that are called bird nest caps. So they're a type of fungus of this group. So if you look at a mushroom, under the kind of cap part of the mushroom, they have the gills. And so if you look at the gills up close, they have these little structures called basidium, which will then have the spores, which are the reproductive structures. Now we have two examples where we have a symbiotic relationship between a fungus and another organism. And our first symbiosis is going to be forming the kind of the organism called lichen. So lichen is going to be something that you've seen growing on rocks and trees. It can sometimes grow on insects, and it is going to be kind of a mutualistic relationship between algae or cyanobacteria and some type of fungus. Okay, and so make sure that when you're remembering your different symbiotic relationships, it can be either a cyanobacteria or an algae, but it is definitely a fungus. So one of the members has to be a fungus. And if you see kind of this image below, it kind of shows how they are going to interact with each other. Here are some pictures of lichens. I think lichens are actually very beautiful, and you can see them when you go on hikes and walks in forests and things like that. The second symbiotic relationship is called mycorrhizae, and this is going to be found between fungus and land plants. So plants are going to have mycorrhizae, or this mutualistic relationship with the fungus that is going to form around the root system. The plants will be able to get water and nutrients from the fungus, and the fungus will then get carbohydrates from the plant because it can perform photosynthesis. And so it's found that plants do much better if they have mycorrhizae on their roots than if they don't. Now, what I wanted to go is just an overview about funguses and kind of the basics of these different groups and some important topics. And so funguses are going to be multicellular heterotrophs. How do they eat? They're going to secrete digestive enzymes and then absorb the food. They're going to have cell walls that are made out of chitin. They're going to be decomposers, which means that they feed off of dead and decaying material. They can be parasites, and a parasite is going to be an organism that lives on or in a host and extracts nutrition from the host. It's bad for the host, but good for the parasite. And a symbiosis is going to be an interaction between two organisms, typically good for both, and mycorrhizae and lichen are going to be good examples of these.